Hi everyone. Today we have James Zhu, who received his bachelor's degree in biology and chemistry from the University of North Carolina at Chapel Hill. He then joined the University of Texas Southwestern Medical Center as a graduate student in 2019 and is currently mentored by Dr. Yang Shi and Dr. Tao Wang at the Quantitative Biomedical Research Center under the Health Data Science concentration of Peter O'Donnell Jr. School of Public Health. He has worked on multiple projects and authored publications involving computational immunology and spatial transcriptomics. Today, he will be discussing how cell-cell communication is essential for how life works and functions, and introduce a multi-instance learning framework called Spacia to detect cell-cell communications from data generated by SRTs by uniquely exploiting their spatial modality. And with that, I turn it over to James.
All right, thank you, Hector. First, I want to thank you for inviting me to give this talk. Uh, and just something, I gave a similar presentation for a committee meeting before, and I was told that my pacing is way too slow. I'll try to move things along, but if you find yourself falling asleep or something, please let me know, and I'll make adjustments as I go. So today I'll be talking about our recently published project on using spatial transcriptomics data to infer cell-to-cell interactions. And this was published a few weeks ago, finally, in Nature Methods. It is a large collaborative project, both within our lab and also with investigators around our institution. And just for the sake of convenience, we focused on tumor samples because that's what's available. When you think about we are all multicellular organisms. Obviously, we have many different types of cells within our bodies, and the communication and interactions between these cells are very critical for various processes. So in terms of tumor, we know that the tumor microenvironment is a very complex place. Most tumors are not just a homogeneous mass of cancer cells. There are many other stromal cells, immune cells, and these play very critical functions in terms of processes like tumorigenesis. That depending on the context, immune cells can be anti-tumor, or they can have some immunosuppressive effects. And here I'm just going to ignore the nuances in the clinical definition between cancer and tumor. But yeah, and so previously the one way to try to uncover this complexity is using single cell RNA-seq. Here I just have an example of a typical analysis using that sort of data. We have some sort of a dimensionality reduction method to try to represent this data in transcriptomic space. Clustering can be performed, and based on the transcriptional data, different clusters can be compared, different treatments as well, to look for differences in transcription. Oh, sorry. Okay, I'll just keep going. One way of looking at it, with regards to cellular interactions, there are some existing tools that takes this sort of data, and using the transcriptional, essentially correlations between the different clusters, they can infer potential interactions between these different clusters. And this is a typical example, just using cell chat as an example. And so in general, these existing methods sort of tend to follow a similar framework. So they take the expression data, really the transcription data from single cell RNA-seq, and that is combined with some sort of database of ligand and receptor pairs. The model itself then tries to fit the data with the with the database in some way. It gives an output of estimated interactions between the different clusters. So two issues here. One is that the analysis is performed at the cluster level. So because cells in single cell RNA-seqs, because there's no location information, we can't really track every individual cell. So most of the analysis generally performed on the cluster level. And because while it's nice to make use of existing knowledge, it also places a limitation on what is possible to get out of these models. But even though some of these databases can be quite large, it's not possible to cover everything, every potential interaction that could occur, because we don't actually know all the interactions that could possibly occur. So in this sense, we're limited by existing knowledge. So with our model that we call Spacia, we decided that maybe we can make use of this, the extra spatial information from spatial transcriptomics to model this process of interaction from scratch. So we get rid of the existing knowledge. So we discard the ligand receptor database, and we build a model that just uses the spatial distances and the transcriptional information from the data to model interactions only using those information. And because of certain unique aspects of our model, we are able to estimate cell-to-cell interactions at a single cell resolution. Because we are not limited by any database, we can look at any genes we want to. So we are potentially able to discover new interactions that were not known in literature. And we can, due to certain attributes of the model again, we can go back and examine our fitted model to estimate the spatial and transcriptional properties of these interacting pathways. So like I think Hector mentioned in the beginning, our model is something that we call Bayesian multi-instance learning. And so before I go give a quick overview of the model structure, I just want to briefly mention what is multiple instance learning. So a typical classifier, for example, we would put in some sort of input, and then the model would then give out some output. So classifier would be true or false. So the analogy here is that I have a classifier that can predict whether a key can open a lock. And when we convert that into a multiple instance learning problem, instead of doing predictions on one key at a time, we put in a collection of inputs. So this collection is referred to as a bag. The individual inputs within this collection is referred to as an instance. So instead of predicting whether one key can open a lock, we're predicting whether a key ring with multiple keys can open a lock. Essentially, if at least one key works, then the model will output true. And this is a more realistic representation of what's actually happening. We don't go around carrying single keys. We tend to have a key chain of multiple keys. And so we decided to think about cell-to-cell interaction within this sort of similar framework. So now instead of key and lock, we're looking at potential sending cells and potential receiving cells. And instead of doing this one by one, so thinking about cells as sort of two individual pairs of these cells existing in vacuum, we know that cells are, in reality, all of them are there the whole time, and the interactions could be in this multiple to one form. There's also like one to multiple interactions, but you can sort of see that we decided to adapt this multiple instance learning framework to look at the problem of cell-to-cell interaction. So this is just the general structure of our model that we call Spacia, which stands for spatial cell-to-cell interaction analysis. And there's much more detail in the paper, and of course supplemental. So here we use the spatial information from spatial transcriptomics to essentially determine which cells are eligible for interaction in a given collection. So in this sort of a collection of one potential receiving cell and the surrounding multiple potential sending cells. And then that information is combined with the transcriptomic data to together estimate if there is an interaction happening. And the interesting thing about our model is that because we use a Vizgen method, we sample these coefficients from the non-informative prior distributions, we can then go back and quite relatively easily look at the properties of our inferred interactions. So here, I illustrated two different types of interactions. One in these sort of purple cells is sort of a contact-based interaction that occurs over a relatively short distance with genes that are positively correlated in transcription. And then in these green cells, we have a secretory interaction happening at slightly longer distance with genes that are negatively correlated between the sending and receiving cells. The interesting thing here is that we don't have to put in some sort of arbitrary cutoffs for distance, which is something that we saw with some other software using spatial data. So ideally, the model itself would sort of figure out the spatial relationship of different types of interactions. We also don't need to worry about the direction of correlation between the gene expression. Again, that's something the model I think figure out by itself. And I mentioned before, we're using the distance between cells to determine sort of eligibility of interaction. We can take that distribution out from our NCMC process, and then we can give a what we call the primary instance indicator for each individual cell. So that is what we use to get at the single cell resolution interactions. So first, we did the initial tuning of the model using simulated data. So here we simulated a bunch of different cells. Assigned some of them to be potential sending cells, assigned others. So here in red as potential receiving cells. Then we have the model predict potential interactions, which we call cell-to-cell communications. And so here, like I said before, the primary instances are the cells that are predicted to truly interact. And so here we have the ground truth, so we know that we're able to show that our model is correctly predicting which cells are actually interacting. We also added in a bunch of noise pathways. So in terms of the gene pathways, we put in 90% of them to be not actually interacting. We have a small fraction of true actual interacting gene pathways. Some of them are negatively correlated, some are positively correlated. And the model is able to distinguish between these. And so next we moved on to actual data. This is a prostate cancer data that was made public by MERSCOPE. So like the previous software, we're able to plot this circles plot of interactions, except of course we are looking at interaction at the single cell level, so we have a much more detailed plot. And because we have the spatial information, we can also go back, plot those back onto the original data. So these interactions are labeled by these little dark arrows. And so this is a bit of a tricky part because we don't really have the ground. Oh, sorry, we don't really have the ground truth of if there's an actual interaction in these real world data. So we have to come up with other ways to compare and validate our results. So we first compared the results from with that existing software. And one thing that we found, and something that we found previously as well, which is part of the motivation for this project, is that a lot of the existing models have a tendency of outputting similar results for things that should be quite different. So for example, 7DE outputted more than half the results are identical between CD8 positive T cells, mast cells, and endothelial cells. So these are predicted interactions with tumor. And we see that some of the others, there's a high degree of non-specificity when it comes to the results. Sorry about the colors here. It's hard to fit in all of these. But with our results of Spacia, we see that most of our predictions are unique to a certain cell type. Some are shared in two different cell types, which is logical since we do have some similar cell types within the data, such as different types of T cells, certain other kinds of immune cells. So we expect some degree of overlap in the interactions. But once the number increases, we have very few interactions that are shared in 3 or more different cell types to tumor, whereas a lot of the existing software tend to output a lot of the same things for different cell types. We look at the pairwise overlap in terms of results. If we do a hierarchical clustering, we see that similar cell types tend to get grouped together, which makes sense. B cell, endothelial cell, and fibroblast are something that I'll talk more about later. And as a comparison, we have cell chat, which is actually one of the better performing software when it comes to looking at this sort of result. But they have a much higher degree of overlap between the results from different cell types, and some of these really doesn't make biological sense. So for example, B cells sharing a lot of results with T cells. And next, we decided to look at epithelial mesenchymal transition, FMT. This is the rather important process when it comes to tumor because vast majority of tumor are epithelial in origin. However, if they take on mesenchymal characteristics, it comes invasive, less apoptotic, and there's also potential for being generally less differentiated. So all these correlate the worst outcomes when it comes to cancer. So here we decided to take the known genes that are markers of KMT and then look at what are the genes in the surrounding non-cancer cells that are predicted to interact with these genes in the tumor cell. So as expected, We found that fibroblasts and endothelial cells seem to contribute to this EMT process. But something that we did not expect was that B cells seems to have a significant contributions to this. And again, because we have spatial information, we can summarize these processes into what we call EMT activation potential, and then plot that back onto the spatial data. See that fibroblasts and endothelial cells have the somewhat different, but also relatively similar distribution of this activation potential, whereas B cells have a significantly different spatial distribution when it comes to EMT activation potential. Another way of trying to validate our results is to look at known interacting processes. And we know that tumor cells can express these immune checkpoints to inhibit immune cells. So PD-L1 in particular is a known immune checkpoint against T cells. Well, we validated using our GeoMx data provided by our collaborator in breast cancer. We found that these two genes, in particular GATA3 and BCL2, in CD8 T cells, it seems to be negatively regulated by tumor PD-L1, and these make sense because these are known to be play roles in proliferation and anti-apoptosis. With tumor cells start having a lot of PD-L1, then it sort of inhibits these pathways in T cells, which reduces the anti-tumor immune response. And again, this B cells started popping up. So coincidentally, the data we have was relatively abundant in B cells, so we decided to also look at what are the potential targets of tumor PD-L1 signaling in these tumor B cells. And we found IDO1 and PDGFRA seem to be upregulated by tumor PD-L1 in these B cells. So there's a lot less data, a lot less existing studies when it comes to B cells in tumor. IDO1, there's some recent result that suggests it could play an immunosuppressive role when expressing B cells. PDGFRA, there's basically nothing in the literature, but we did find in the other single cell data that we have with the immune checkpoint treatment, PDGFRA B cells seem to go down. So this sort of supports our results that there is some sort of interaction between these two things. And next, we expanded our analysis to different types of MERSCOPE data. So they kindly provided data on different types of cancer. And the hope here is that the findings Spacia has on these samples are able to be generalized to similar cancer types in larger data sets. So here we predicted this PD-L1 signaling, PD-L1 interacting genes in CD8 positive T cells for a number of different types of cancers. And then we looked at the expression of these gene sets in the TCGA dataset. As you can see, these gene sets are highly rich in patients with high PD-L1, so that sort of cooperates our finding. And next, since we saw this enrichment in our genes that we predicted to interact, we wonder if we can come up with some sort of a signature that is relevant for patient survival. So counterintuitively, when we just dichotomize by tumor PD-L1 expression, we found that higher tumor PD-L1 appeared to be correlated with better survival in patients. So there are several different possible explanations. One would be that it's indicative of of the presence of a immune response, which would be positively correlated with survival, despite PD-L1 being a checkpoint, which inhibits the immune action on the tumor. So here we won't delve into the specifics of this mechanism of this difference. So we just want to look at this our predicted PD-L1 interacting genes make a difference or have a better signal than just tumor PD-L1 alone. Because we predicted these genes, the prediction that we made were that these genes in CD8 positive T cells are responding to tumor PD-L1. So we also divided the patients by their level of CD8 T cell infiltration in the tumor. So if we look at these two plots, that those with low signature and high T cell infiltration have a significantly worse survival than those with high signature and T cell infiltration. However, when there was low CD8 T cell infiltration, our signature is not able to distinguish between better and worse survival, which makes sense since our signature is supposed to only work in CD8 T cells. And we look at breast cancer, we see a bigger signal, but again, if there's no T cells, then the signature doesn't really make a difference. And overall, our signature is more predictive of patient survival than just looking at tumor PD-L1 alone. And again, this is essentially a different representation of the data. Higher signature is indicative of lower signature is indicative of worse survival if there is T cell infiltration in the tumor. But this is accounting for the different types of cancer. And in breast cancer, we account for different stages. We see the same thing. And one of the final validations that we have was looking at the role of gamma delta T cells in liver and healthy liver and tumor-bearing liver. Gamma delta T cells are generally a smaller population of T cells. So most T cells are alpha beta. And so they're named after the components that make up their TCRs. However, in the tumor, gamma delta T cells are relatively enriched. So we wanted to see what are the roles that it's playing in tumor, in the liver. When we did our predictions, we found that compared with healthy liver, in liver cancer, gamma delta T cells appear to have a lot more interactions with the hepatocytes, and they tend to be spatially closer to these to the liver cancer cells versus the healthy liver cells. And when we look at the specific interaction pathways, see that in healthy liver, the gamma delta T cells interact mostly through different metabolic pathways. However, in liver cancer, it is interacting with these cancer cells mostly through antigen presentation, and apoptosis pathway. So for our future plans, we want to explore those interesting novel findings a bit further. So after talking with their collaborator, there's a lot of interest in specifically looking at the role of B cells in tumor We know they're there, but it's hard to say what exactly they're doing. But based on our results, it seems to suggest that some of these are potentially playing a suppressive role. But it'd be more interesting to find it, to explore that further. And of course, the gamma delta T cell results in liver cancer that I just showed. This is a relatively understudied portion of when it comes to T cell. Of course, there are other tissues where gamma delta T cells are enriched, things like prostate cancer. So it'd be interesting to look into that further as well. There's also interest in looking at things other than tumor. So there's more to life than cancer. I think immunologists in particular would be quite interesting to see what are the complex interactions occurring in the lymph node, for example. And finally, we want to further improve the quality and usability of Spacia itself. I think we mentioned in the paper that we could look into using Bayesian lasso to do essentially automatic feature selection. So instead of using different filters or existing knowledge, we can just run everything and ideally have the model output the relevant interactions with less user intervention. And there is still some performance areas with performance with Spacia that we'd like to improve, to make it run faster, make it easier to run for everyone. And any questions? I think I went a little faster than I expected.
Thank you. I think that was wonderful talk. This is the last slide.
Yes.
Perfect. Thank you so much for this wonderful talk. If there are questions, please feel free to unmute or post on the chat.
This is kind of interesting. Okay, so I'm a biologist. Okay, so I don't know whether you can see that, but I look at the microscope on an everyday basis. So understanding these cell-cell interactions is very interesting. And the question basically comes up is, a lot of these postulated relationships, how do you know they are one-way, two-way, multi-directional or not? Any thoughts on how you go about trying to understand and validating some of these relationships? Because all cell chat, cell phone, and all that things does is postulate relationships, right? How do we know that they really exist? What the nature of it is? Any thoughts on taking a deeper dive in terms of molecular mechanisms there?
Yeah, that is a very difficult question, actually.
I know.
Because
why should I make your life easy?
yeah, so there's some fundamental limitations here because so for example, the previous software that uses single cell data, right? There only thing we have is just transcription, right? So they have this database. They know that these proteins interact, but if the data is only how much of this is present in this cell, how much is it in this cell, then at some level, right, you're basically saying that if the expression of these two known interacting proteins are correlated, then there is an interaction, which we think it's not a very sound logic from the biological perspective, right? It doesn't necessarily, like, if you have a higher expression of the ligand and higher expression of the receptor, that doesn't necessarily mean there is an interaction. So for our method, we decided, that's one of the reasons we decided against using sort of a database approach. So ideally for us, we want to look at something upstream or something that's secreted or the ligand itself, that's okay. And then we want to look at something that is downstream of the actual receiving protein, so the actual interacting protein. So instead of looking at the proteins that directly interact, we want to look at things that are regulated by these proteins. So maybe some things are upregulated, some things are downregulated. So that's sort of our
So
our logic.
let me back off a little, and you know, I'm not trying to put you on the spot, but I'll tell you why I'm asking the question. Okay, because the classic biomarkers, let's talk about, say, HER2 on the cell surface, right? We have 100 patients treated with anti-HER2 therapy. Only a proportion of those respond. So why do a proportion not respond, and why do a proportion respond? Obviously, the cell signaling is what's going on. So so when you're doing this spatial analysis, can you kind of throw some light on not only the presence of those ligands, but downstream pathways, as you pointed out, and try to help us biology people in trying to understand what could be the possible ways and means that are occurring at a tissue level, because anything can happen in a cell line. And that's the problem with working with cell lines. So we really want to know what's happening at the tissue level. So trying to understand, and you know, we are looking to you guys to give us, help us with the answers. So I'm not trying to put you on a spot and say, hey, your work is not great. Your work is good, but I'm trying to push you into doing something that will help us further. So when you're looking at those interactions, how much of downstream events can you study? The more you study, the more greater is the fidelity of that interaction. So that's really where I'm coming from. How much insights can you provide? And are you looking into those kind of things? The PDGFR, PDL1 was a great kind of a story, but can you expand more on some of those things and give us some biologically actionable reasons as to why some patients respond and others do not? That's where I'm coming from.
Yeah, I think that is something that we are very interested in as well, because our hope is that if we essentially what we can do here is to just put everything into the model and then see what are the possible interactions. So because we're not limited by the database, right? We can just put in everything. Of course, there's a cost in terms of computation and time, but then it's a little hard to interpret the results as well, because I guess that's one reason the previous software are sticking with the database approach, because we know there's a clear rationale that these proteins are known to interact. Whereas if we, even though from like a signal to noise perspective or biological perspective, it makes more sense to look at these more downstream genes. The relationships is very indirect, and of course there's always false positives. There's probably some false negatives. I think we may have erred a bit more on not having as much false negatives. But in the end, we have to have some sort of heuristics to just try to determine which of these are likely to be the true pathways, which are not. But we end up with data that is a bit hard to interpret, because there might be a lot of different layers between our sending genes and our receiving genes. So yeah, that is a difficult problem. The one way we try to do that is by pulling things. So that CDA PDL1 signature thing is when we basically took a selection of the best results, and then we sort of integrated all of it into one signature. So that's a relatively simple approach just using that signature to try to predict survival. But looking at things more deeply, I think it goes it might be beyond just a computational approach. We'll have to do there has to be more like a experimental validation of these things.
Yeah, totally. And I'm not saying that, but if you can provide leads for us, then we can look at it in the experimental models.
Yeah.
I think there needs to be synergy between what you guys can do, because we cannot, and try to understand and help each other out in terms of understanding what's going on. So I don't want to dominate the conversation. There may be other people having a question, but I really wanted to put a plug in for working together. And that's the reason I'm on this call, trying to see how much we can learn from what the work that you and other people are doing.
Yeah, I see. Because I actually have a experimentalist background, so I totally understand the thinking here. And
Wonderful.
yeah, definitely there's I see these especially for this type of software for predicting interactions where it's really hard to validate these things.
Okay.
It's really like a source of like an initial screen for possible hits. So it sort of provides you with something to work with versus something that's like very highly reliable. That's the way I sort of see things.
No, thanks. Great talk. Amit, who else has a question or comment?
Wonderful. No, I think we are enjoying the conversation. Are there anyone else who had a question? Please feel free to unmute.
Hi, James.
You're good, Surgeon. Yeah.
Yeah, thank you I put in the Q&A, but it was not picked up. But a few things, just like a technical questions, I did not pick up. So to implement Spacia, do we need to know the cell types prior, or like do the cell type annotation to implement Spacia, or just the expression matrix or the count matrix is enough?
Yes, so what we did is we did the cell typing beforehand, but you know it's not the model is not, in theory you could just assign some arbitrary cell types, so you don't necessarily have to have like the most well-defined cell types. So as long as you can identify two populations that you want to know what sort of interactions is happening, then that's fine. But in general, it makes more sense to actually do the cell typing and then look at interactions between specific cell types.
Okay, thank you. Another thing is like some data which you show, I came across is MERSCOPE or something. Do you think it can be applied to like spot resolution, like 10x Visium data, or it's only for single cell resolution
Yeah,
platforms?
for us, we are really just looking at single cell spatial resolution data. So the methods with multiple cells in an individual spot like Visium probably wouldn't work very well for us. We know that there were some, not some, actually several other slightly earlier methods that they seem to be really designed for Visium data. Those tend to have performance issues if you put in single cell resolution spatial data.
Okay, thank you. And my last question is, any reason, like when you're doing the comparison with the standard platforms of softwares available, like I didn't see NicheNet, because when you're saying you're trying to see the going from the ligands to directly the downstream of receptor and not just ligand receptor, is there a reason why NicheNet was not chosen for comparison?
I think we did some comparison with NicheNet. It's probably somewhere in the supplemental. I think
Okay.
I did that part. For the other, I just basically I just picked the more well-known ones, the ones that ran well. So there's not really we don't really have a good explanation of why I didn't put NicheNet in the plots. But we did some comparison, I think, but the data is not as readily available. But yeah, I think if you dig through the supplemental enough, you probably find something on NicheNet.
Okay, thank you so much.
Thank you, Singh. Are there anyone else with a question?
I have a question.
Yeah, go ahead, Daily.
First of all, thank you for the very interesting talk. Thanks. I just have one quick question about the method that you're using, the Monte Carlo, Markov chain Monte Carlo method. Could you explain on why, like specifically you choose that method and like just the logistics behind it? Thank you.
So that's an interesting question. So this will go. This kind of goes back to my PI, basically. So we developed a similar method, using the same framework. The idea there is to simultaneously solve a multi-instance learning problem, as well as try to infer which of the instances in there is responsible for what we call the primary instance. So as a part of that project, we had a collaboration with a statistics professor over at UT Arlington. So I probably can't answer the if you want the statistics, I probably can't really do that on the spot. But it is in there. We have a reference to that paper as well. It's a slightly different form. And essentially my PI Tall, he always had these crazy ideas, right? And surprisingly often they work quite well. So basically he says, well, we have this really innovative statistical method that's good at solving these complex issues. What other issues can we apply this to? And then sort of, spatial data was very hot during that time, still is. And then basically thought of a way to apply that model to the problem of cell-to-cell interaction. And then we can, so the original model, they use two sources of information to run that model. So when it comes to spatial data, then we decide to use the spatial and transcriptomic portions of the data as that two source of information and combine it all into this particular model. And luckily, we found that it worked pretty well. So yeah, there's not it's not as beautiful as we looking at a problem and then coming up with sort of a perfect solution for it. So it's sort of like we have this tool and then we found a problem that seems suitable and then we tweaked the tool a little bit and made it work in an interesting way.
Well, many discoveries are made that way, so no shame in that. So good work, though.
Wonderful. Are there any other questions? If not, let's. Oh, we have more. Yeah, go ahead.
Sorry. Yeah, so hi, James. Thanks for the nice talk and thank you for the wonderful conversations as well. I'm wondering, so you went from the previous cell-cell communication method that's mainly detecting the non-regulatory pathways to this multi-instance learning, where you can, it seems to be like detecting more in a unsupervised way, trying to find the like
find the cell-cell communication in a more unsupervised way. Is the natural next step deep learning, or are there already current efforts in this field that's trying to apply deep learning methods on detecting cell-cell communication? Thank you.
Yeah, that's a good question. Actually, there are already a number of methods that uses deep learning to look at this problem. We, in particular, we didn't really like doing it that particular way because deep learning, the ones that perform well tend to be difficult to interpret. So I think one major advantage of our approach is that it's relatively easy to interpret the model. And so for this particular problem, we want to go back to the model and then look at aspect of the model to infer properties of the interaction. So I think that's one of the major reasons we didn't go with the deep learning approach. Not to say that it can't work. It's just that sometimes statistical approaches can offer certain advantages, especially when it comes to interpretability. And also, if you have a strong biological basis, so you have some sort of clear indication of what's going on, then sometimes a simpler model can actually perform better and it be easier to explain. But yeah, there are deep learning based methods. I think there were some things looking at spatial neighborhoods. Can't think of the name right now, but very well known. But yeah, they do work very well, but there are advantages and disadvantages.
Thank you. And also wondering, what are the major criteria people benchmark their different methods on regarding the cell-cell communication? Are there any?
I feel like there's not really a very well standardized criteria. So it's kind of like everyone just doing their own thing. Fundamentally, there's not a the real big issue. There's not like a fundamental ground rule that you benchmark everything against. So for us, we try to show that we're not as non-specific, and that we found things that are biologically logical. We found some big things that are interesting. So that's essentially our way of benchmarking. But yeah, the benchmarking is really not very well developed for this particular problem.
Thank you.
Thank you, James and Cheryl. And are there any questions? If not, let's thank to the speaker again.
Thanks.
Congratulations. This was a great talk. Thank you, James.
Thank you for inviting me and giving me this opportunity to talk.
This was great, on time and very detailed, and appreciated taking the time. So with this, I think I'd like to also announce that next speaker actually also will be on inferring the patterns, pattern driving intracellular flows from single-cell spatial transcriptomics from Ching Ni's lab, UC Irvine, and his name is Axel Almet. He's a postdoc at UC Irvine. So we'll be having him in 2 weeks. Please stay tuned. And thank you everyone for participating today, and hope you guys have a wonderful weekend.
Take care.
Thank
Bye.
you.